And as you heard also, the meeting is being recorded. So the vision of the Life Science Cluster is to support sustainable industry development within the areas of life science. And as you know also, this covers both health, marine, agri, industrial sectors. We would like to create and develop more jobs also through strengthening both collaboration and knowledge sharing between and within the industrial, academic, and also the private and public sectors. And this event is definitely within the scope of what we would like to do. So we really look forward to see how this initiative can grow further. The cluster today has more than 90 members and spans both large multinational companies and also small companies incepted this year. It also includes universities, hospitals, institute sector, investors, and other types of players. And it's a good community to also initiate new ideas. This is also a pipeline of the approximately 35 companies that are in what we call investable stage. And hopefully also from your initiative, we believe that many of these companies will have use for the technologies and the new insights one can gain from using technology in new ways. And also developing solutions for society through science, and also then unleashing the potential of creating new solutions on the market to the best for society, for patients and others. So by that, I would like to hand both the word and the scene over to Thibaud, who will guide you through the rest of the program.
Yeah, thanks, Mette. Hi, everybody. Very excited to see so many of you on the welcome we had with every people we spoke with. Also have my slides, so I'll just go on just to present you actually the community on what we aim to do. So the name is not decided yet, so that will be one of your first tasks to do, but I would like just to come back to see how we came up with such initiative to build a community. And to know that, I will just go through like who are the funding teams that make me more clear. So I am the initiator. Actually, I'm used to be a biologist because I have a formal training as a structural biologist. But at the same time, I've been using PyMol for more than 10 years. And I merged both the biology and the informatics during my PhD at the University of Tromsø when I applied molecular modeling to find new therapeutics. And then moved to Oslo to work with Michele, Cascella, that will be presenting afterward. And I was surprised to see that the field of molecular modeling was not developed in Oslo. And when last semester, after some exchange with Professor Trond Vidar Hansen, he has been advocating for the School of Pharmacy to develop such competencies. The idea to organize a seminar in molecular modeling started to merge in my head. And through the different collaboration, and as I'm as well partner or member of the Life Science Cluster, I've been put into contact with several people, and we put together a group in order to organize this seminar. And then it grew, the idea grew, and then we end up as a committee. And now we are 4 of us. So it's myself, Professor Trond Vidar Hansen from the University of Oslo. It's also Dr. Naji Akok, which is as well, he has 2 adjunct associate professor positions at the University of Oslo and similar. And he's as well the CEO and the co-founder of a small startup called Invirtualis. And the last one is Richard Norman, which is actually ex-AstraZeneca. And as well, he has found his own consulting company, which he is now actually working with BioExcel, which is a center of excellence, and they will be presenting during today's seminar. So the vision we had through our different meetings was that we want to build an active community with everybody in Norway that has an interest in molecular modeling, because we have seen that it is a fragmented field. So we need to gather everybody under the same umbrella. And at the same time, the field of life science in Norway is not mature yet. So they still need to develop the awareness of such field and the technique that can be used. And doing that will then increase the Norway attractiveness at the international level, because Norway has a lot of possibility and we want to reach to that. And in order to reach to this vision, we have seen different activities that we should do or we could do, and so we want first to develop local competencies. So it can be through like formal or informal meetings. So it can be some meetups, some seminar, workshop, or even some congress. We want as well to provide access to resources. So it can be software or hardware. So for instance, with Excel, or we can have access to some cloud computing, GPUs, IPUs. So if you need, as I mentioned, we want the community to be active. So it's for the community, by the community, and we support each other. Because I think one of the key here is to have a secure and safe environment that you are not afraid to come and ask questions, and even you can find your next collaborator or a new friend and so on. And last but not least, it's like we want to be a hub for innovation and collaboration. And we believe that if we share knowledge, not only about the science, but as well the funding possibility, and then we want to advocate for collaboration between industry and academia. And the first task that we're going to ask you today is to think about the name that you would like this community to take. So we have this NorCamD or CamD Norway. We can decide that later during the feasible discussion. And also it's like, what do you expect or what do you need from such community? We need you to tell us how you want the future of this community to be. And as well, what kind of activity you would like to have. Is it more seminar? You want more knowledge? You want like workshop? Tell us. We need to know. And without further ado, I will stop sharing my screen. And we're going to move on to the first keynote, which is Professor Michele Cascella. He is a professor at the Hylleraas Center, which is the Quantum Molecular Science, and at the Department of Chemistry at the University of Oslo. Michele, the stage is yours.
First of all, thank you for the invitation and for organizing this very interesting event. Of course, I was tasked to sort of introduce the field in a sense, so I will not really present directly most of my research, but I wanted to have at least for those of you that don't know me, just a very brief resume of what I'm doing in my lab. So I'm interested in simulations, so multi-scale level, of typically especially for this community, I think the interest is in my research in nanoscale self-assembly, and we work both with proteins and lipids and surfactants. And of course, we are now particularly active in method development and the multi-scale modeling. I will briefly touch this towards the end of my seminar. But stepping directly into these particular topics, so clearly, why do we want to do molecular dynamics? What is molecular dynamics and what we can do with molecular dynamics? So first of all, I would try to make a step back and say, what about modeling? Why we are interested in modeling, particularly molecular modeling? Clearly, we know that life and anything surrounds us is built in a multi-scale level. It's like you start from molecules, you build macromolecules, and then you move up. So it's like in a microscope where you zoom in and out, you can just go from jump scales and change the object you observe. Now, when you are at, let's say, larger, more complex systems or larger bodies, we typically use phenomenological ways of modeling or understanding or interpreting the reality. But the nice things and the magic, the beauty, the elegance of the nanoscale, so the scale of the atom and molecular level, is that at that level, the fundamental equations that we use to interpret the realities converge. So all the systems at molecular level independently of the larger, let's say, assembly they constitute, we respond to the same universal laws. So we have a universal way of interpreting the reality that comes clearly from quantum mechanics that allows us to build a systematic microscopic to macroscopic bottom-up strategy that also allows the interpretation of also all the possible interference between different systems. For example, if you are interested in nanomaterials or nanobiomaterials and biomaterial interfaces, and that normally we describe the bio and the nano and the material with different phenomenological equations at the large scale. At the microscopic scale, they will simply be described exactly in the same way. So now, what is fascinating about the Schrödinger equation, which is at the base of everything, is that it is indeed it's a time evolution equation. So it's the time evolution of the wave function. And in general, most in general, all systems, all complex systems evolve in time, so are subject to some equational motion. And if we are able to follow the motion, understand and interpret these equations and solve them, we can in principle study everything. And then every system has its own equation. So it could be a molecular system, but also meteorological system, even markets, economic markets. So there is a universal need to describing systems in time evolution. Now, I want to face immediately what is the problem. So what is the problem? And I will simply use some videos from YouTube. This is basically as a very simple, mechanical system formed by 3 pendulums that are attached to each other, and they are left to oscillate. What you don't see is that this is not one pendulum, but these are several pendulums that are actually separated by 0.01 radians at the beginning. So they, at eyesight, they are identical, but they are not. They are actually superimposed one on each other. Now, what happens is that after a few oscillations, they initially are coherent, they will eventually separate, and they will start having a chaotic behavior. So as you see, after very few oscillations, these 5 pendulums behave like they were. So you cannot recognize that this pendulum started from the same position. So this is just another example. I'll let the video go. But if we see now, instead of few pendulums, we put, and this is, I was lucky to find this video where we have 1 million of pendulums that start from very tiny differences. So you will observe that after very few oscillations, then the pendulum eventually, you will reach these critical points. You start having chaotic behavior, and the pendulum will distribute chaotically over all possible configurations that each pendulum may take. So I let the videos flow for those of you that like this. But the important thing here is that even though we would like to study the mechanical behavior, we are facing this fundamental problem that is known as the atom instability, that basically is telling that we cannot really predict the oscillations of a pendulum, so anyway, or a dynamic system after a certain time because of this chaotic divergence. So what is now the point if we the predictivity, so the reproducibility of the oscillations or anyway of the motion is practically impossible because you need error and machine precision to eventually propagate and form this chaos. So it's then molecular dynamics and not predicting anything or anyway only in a very short window. So so the reason why molecular dynamics is still very successful, in fact, and is very useful to study time evolution, comes from this other video. So now, forget for a second that the pendulum is moving, but that if you have many pendulums, then they will chaotically cover an ensemble of conformations that is now depicted in this snapshot. And now let's see what does a single pendulum do. Now, in this video, you are following the pendulum and you're keeping track of all the configurations that the pendulum is covering. As you see, over time, the pendulum will slowly, even though it's chaotic, will progressively cover all the conformations that are actually obtained by the chaotic ensemble of all pendulums. So this is a fundamental observation that takes the name of ergodic theorem, is that if you let a system, chaotic system, evolve over time, then it will eventually explore all the possible conformations with actually the right probability. So this means that even though the individual trajectory might be every time different because of numerical uncertainties in the initial positions, globally, each independently on the trajectory that you follow, each trajectory taken all together will explore eventually the same configurations with the same probability. Why this is important? This is important because when say chemical level, or anyway biochemical level, biomolecular level, we are actually in a world of thermodynamics. We are in the world of Avogadro numbers of objects. Typically, we have orders of the Avogadro number of molecules and biomolecules. And we always measure, when we are in a lab, we measure statistical ensembles. So every time we perform measure in a lab, we don't measure one molecule in one geometry, in one special geometry, but we typically measure over an ensemble of molecules, and the measurement will take always a finite time, so these molecules will move while we measure them. So every measurement that we perform in the lab is actually a statistical average over all the possible configurations that our system can take, so every possible geometry, weighted with the probability that if we are at thermodynamic equilibrium, this probability is the Boltzmann probability. So if we want, so in the experiment, this is what we have. We have really an ensemble average, so an average based on ensemble of structures. Now, if we want to predict that now in a computer model, you have to be able at the same time to have methods that can reproduce an observable from a microscopic state, like the geometry of your molecule, but also be able to predict all the geometries that your system is possibly taking with the right probability. Now, this is formally a 6-dimensional integrals because you have to sample all the possible geometries of your system. So you have x, y, z, and x, p, y, p, z, so the velocities and the positions for each atom that composes your system. So this is a 6-dimensional integral, and you understand this is numerically impossible to sample directly, so just systematically. And this is where the ergodic theorem kicks in. That it tells us, forget about this complicated integral, just follow your, if you're able to perform molecular dynamics, if you're able to move your object according to the right equation of motion, then you can simply run your system over time and collect your observable over time and average that. Then once it converges, if the simulation is long enough, then you will have reproduced the, let's say, statistical measurement that you perform in the lab. The beauty of all these expressions is that they are formally exact, at least on a statistical level. So in the statistical theory here, we have not introduced any approximation yet. Of course, there will be in any method that you use eventually. But at least formally, these are really very nice results. So the reason of success of molecular dynamics, and this is something maybe counterintuitive, is not in the capability of predicting the time evolution, but it's actually in its success as a sampling method, as a statistical sampling method. So this is the primary reason why we want to do molecular dynamics. We want to obtain, have the possibility of obtaining from a straightforward method thermodynamically converged properties that we can compare directly eventually to the experiment. Clearly, in this machinery, we need an algorithm that performs integration of the equation of motion. And the beauty of this approach is that this is universal. So this is true for any system at any resolution for which you can write a time evolution equation. So it can be true for a system that is treated at pure quantum mechanical level, where you want to solve the time-dependent Schrödinger equation on any or any of its approximations, any classical approximations. But it is true if your system is treated as a classical mechanic system, so with the classical particles that follow Newton equation, because Newton equation is again an equation It's a time evolution equation. Or maybe you are not interested in the detail of the molecules. You want to study, for example, fluidodynamic equations, like the Navier-Stokes equation, for example. So now, of course, the cost of molecular dynamics, so what you can actually do, depends dramatically on how complicated it is to evaluate the different energy and forces that you need to integrate this equation of motion. And of course, how large is your system? So for how many particles you can simulate. And clearly, if you can afford simulating many particles, then these integrals, because they are statistical integrals, will converge more rapidly because you perform more statistics over time. Typically, when we study biological systems, we typically study actually one only object. We study one protein. Otherwise, this will become immediately very large and then very costly. And then this means that these simulations typically have to run for longer if you want to simulate, have converged results. And of course, this depends also on this convergence of this limit, of this average. If your system is fast diffusing, likely you need to sample shorter times. In a slow diffusing events, like in protein folding or in membrane self-assemblies or these kind of systems, you are forced to simulate very long times. So this problem of the cost of the individual action and basically the cost that you have to do due to the largeness of your system has over the times produced a hierarchy of methods that is like, again, it's like a microscope, so a computational microscope that you use to zoom in and out. So the most accurate methods that you treat the system quantum mechanically are very costly, and then you can simply simulate systems in the order of thousands of atoms or maybe a bit more, and in a time that is in the multi-picosecond to the nanosecond time scale. Then, of course, if you move to a classical mechanics formalism, you can simulate much larger systems. You can jump several orders of magnitude, both in size and in time. But for biological systems where these methods may still be actually too expensive, and then especially if you want to study large assemblies or, for example, soft matter, soft biological matter, then you need to lower the resolution to not atomic, so semi-atomic, semi-molecular level. So where you have description of the molecules, but you lose the atomic resolution. And this is also another hierarchy of methods, coarse-grained methods. And then, of course, this eventually evolve into the continuum where you can study larger systems at longer times. So just very briefly, what I would like to give you in the last 5 minutes is just a very brief idea of what you can do. So at the initial molecular dynamics level, again, I said there are several semi-classical approximations that become, let's say, less accurate, but more practical. And clearly, this is not a resolution to which you want to do biology or study biological systems, but you may still need actually large quantum mechanical studies if you are interested, for example, in long-range electron transfer, for example, people that study photosystem 2 or electron transfer proteins, where you really need large quantum mechanical systems to have this long-range coupling between the electron acceptors or donors. And this is just to give you citations. This is the very first protein that has been simulated at pure quantum mechanical level by Todd Martinez. It's a very small protein, very short times, but it's a proof of principle in a sense. At classical mechanics level, so the first approximation we do, this drastic approximation we do, if we are not interested in any properties that depends on the electrons, so no chemical reaction, no photosystems, then the electrons are basically gluing atoms together. But if, apart from, we can represent effectively this gluing in terms of small fluctuations between the equilibrium, around the equilibrium distance, and this can be used by simple Springs models. So we have this classical dynamics methods, or empirical force fields. These are based on mechanical properties, and if you are interested in how this force field have been developed, there is a nice retrospective by Michael Levitt you can read. So we simply describe, in this case, the molecular interaction are described simple mechanical potentials that take into account any deformation or take into account the intermolecular forces by point charges and Lennard-Jones potentials to represent demand and mass exclusion. So this is the typical workhorse of molecular modeling, at least accurate, let's say, molecular modeling of today, and you can study the resistance from polymers to membrane proteins to DNA and so on. And again, just to give you an idea of a brief idea of the basically some landmarks in simulation, this was the very first paper where the group of Klaus Schulten touched the million of atoms, simulation of a million of atoms, studying the full satellite of baculovirus. Imagine also the solvent is also an atom, so it's also explicitly represented. Then they had another landmark paper in some years ago when they studied the viral capsid crossing the 10⁷ atoms. And now recently they published another paper with vesicles and synaptic vesicles with, again, another landmark. And this is a simulation by the group of the show, where they, for the first time, a group managed to cross the threshold of the millisecond. Now, of course, they have many other studies over the years, but this was really sort of the landmark, the first, let's say, crossing of the millisecond timescale. So, of course, as this is a method that doesn't allow you to have chemistry, so very prominently, Nobel Prize 2013, is the use of the combination of ab initio molecular dynamics and ab initio methods and classical molecular dynamics with the QMMM. There are many different flavors of QMMM. I just present the one by Ursula Rotlisberger in EPFL, where couples really the Carparinello Hamiltonian, so you can do really ab initio molecular dynamics within a classical mechanics model. You can really simulate, for example, photodynamics in a protein. Now, just very few last slides. Coarse graining. It's a method. There is this idea of zooming out with the microscope. So we go from a chemically resolved molecule to a pseudomolecule that resembles, so where bodies in these pseudomolecules represent groups of atoms. So you have a structure that resembles the structure of your peptide, for example, but you have lost the chemistry. The advantages here is that you reduce the number of bodies, they are cheaper, the effective potential you have to use are typically smoother, so the dynamics accelerated, you can simulate longer times more easily. It's very good in describing multi-phase systems, so this is today becoming the workhorse to study, for example, lipid assemblies. The disadvantage is that, of course, there is lots of chemical information. If this is something where you are interested, then this can become tricky. And of course, this procedure of coarse graining is not really well defined. So we have the problem of deciding how to do it, and this is still an open problem. And so there are basically some models that may perform better or less better according to the properties you want to simulate. But this is a work in progress in the community, and this is just to give you an idea what we can do. This is just a recent study where we are studying, for example, self-assembly of surfactants and the effect of, for example, of non-salt in charge surfactants. So you can see the formation of progressive formation of spherical phosphorylated to cylindrical micelles in the video. So we have also studied, for example, self-assembly of bilayer and complex lipids like lipopolysaccharides and things like this. And, just to give you a flavor of what you can do. Now, in the last few minutes of my seminar, I would like to talk about what you cannot do, or at least what is hard, when molecular dynamics fails. And this is typically, so the ergodic hypothesis tells us that if you keep running the simulations, you will explore eventually all conformations. But when you are in a situation where you have an activation process, like in a reaction or anyway, any system that requires activation, then the probability to go over a transition state decays exponentially with the energy. And then what happens is that you remain trapped in one configuration. So activity processes are what we call slow processing in a lab. So if we have a reaction and it's a slow reaction, you will see a continuum, a slow continuum change in color, for example, while the reactant produces the protons. Now, this is a molecular level, this is not a slow process. This is an unlikely process. So it's probabilistic. So we do not move the molecules do not slowly move from A to B and to C and D, but they will remain A and B for a very long time. And then if they win a lottery, then eventually in a very short time actually that. So now, if you have an Avogadro number of molecules, then you can always have the chance of winning the lotteries and then see the reaction slowly progress. But if in a simulation you have only one molecule, then you will clearly not win the lottery. These processes, so a creative process in molecular dynamics simply fail. You cannot do that. Now, in order to do that, you use enhanced sampling methods. So these are family of theoretical approaches based on statistical mechanics. They are effective. They can help you sampling all the states and also reconstruct the free energy profiles. The problem is that they still use molecular dynamics as sampling, but these are computationally demanding and not black box. So while molecular dynamics today is becoming straightforward, enhanced sampling methods, you have to really have hands-on and be proficient in your lab to do that. So where to go for the future? Clearly, the ultimate goal for molecular dynamics, now the field is becoming very mature. The ultimate goal is to push the boundaries in order to reach characteristic times and sizes for the experiments, so that we compare directly what is seen in the experiment and what is seen in the computer. And for that, we need continued development hardware, exploring new computational facilities like exact state supercomputer being built in Europe in these years, continuous development of software, and continued development of models and methods. Because as I show you, the cost depends on the model. So if we can improve the models and make them more effective, then we can push the limits also of molecular dynamics. So the other problem is that the larger system, the longer times we simulate, the more junk data we produce. So we produce a lot of noisy data. We can easily obtain terabytes of data for long simulations. So we need a combination of data science and machine learning to help molecular dynamics today, in order to both build models, machine learning, to build better models, and improving the transfer for accurate, very costly methods to maybe cheaper but less precise methods today, and computational data science that can help actually reduce the noise to signal in our in data analysis. If you're interested in knowing how to do what is being done in machine learning, I suggest this publication. This is just 1 or 2 weeks ago that they came out. And actually, I made some advertisement. Arturo Guzik will be invited in September. Actually, he's invited, accepted to be the next Adolf Gluckman lecture. It's a series of honorary lectures that is in our center. So for those of you that are interested, follow our website, because he will give a seminar in September. And this is open to the public. Okay, I just leave this slide. I will not comment that because I'm out of time.
Yeah.
But I'll just give this slide as auto provocation. Okay.
Okay. Thanks, Michele, for this broad overview and as well for your insights on. I would like now to give the floor to Dr. Richard Norman, which is as well the managing director, as I mentioned, of Norman Consulting, but he's as well a partner in BioExcel. And together with Dr. Rossen Apostolov, which is the BioExcel coordinator, and from the Karolinska Institute, the Royal Institute of Technology, and they will present BioExcel toolbox, advanced application for molecular modeling simulation.
Yeah, thanks very much, Thibaud, and everyone. Thanks for inviting us. So as Thibaud said, this is a joint presentation. I will go first. It's going to be a whistle-stop tour of BioExcel. So by way of background, if you can move on to the next slide, Rossen. Rossen's driving the slides for us today. So I don't think I need to go through my profile. Thibaud has already introduced me. So Rossen is based at the KTH, Royal Institute of Technology in Stockholm in Sweden. He's the BioExcel coordinator, and he used to be a developer of the molecular dynamics engine GROMACS, and we'll talk a little bit more about that later. I'm based in Norway, and I used to work in the farm industry, and I'm a structural biologist by training. So if you can move on to the next slide, please. So the BioExcel Center of Excellence for Computational Biomolecular Research, it's a project. It's funded by the European Commission under the Horizon 2020 program. We're currently in the second phase of 3-year funding, and that ends this year. There's an upcoming third phase, which is expected soon, and this will be coming out under the new Horizon Europe program. the mission of the center, and I use sort of center and COE interchangeably throughout the presentation, so just wanted to warn you there. But the mission is to develop high quality user-friendly software for researchers in the field, to increase user expertise and skills, and to strengthen the relevant community. And I'm going to just tell you a little bit more about how we're doing that in the next few slides. So next slide please. So our codes and expertise are focused on everything from atoms up to cells, and thus they're relevant for biochemical and biophysical studies, including the studies of lipids and membranes. And also importantly, they can be applied to the discovery of various sort of drugs using different modalities, whether this is small molecules, peptides, or even antibody-based therapeutics, and there are others as well. Next slide, please. So we are a COE, we are a center of excellence, with excellence being the operative word here. So we bring excellence in our solutions and excellence in services and support, and therefore we increase excellence in usability, and therefore open the core codes up to many more people in the community. I think we can confidently say that our strength is in developing high quality state-of-the-art software, and in the last few years we've been able to make this much more usable and accessible to users in general by providing the support and training that's required for this wider adoption. And this is not just to academic users, but also to industrial users who already have access to commercial tools and services. Next slide, please. So our core codes, they're open source and they're free to use. These are the ones that we support. So GROMACS for molecular dynamics, Haddock for integrative modeling, and PMX to set up free energy calculations in GROMACS, and also CP2K for hybrid QMMM simulations. And although we support CP2K and we've created an interface between CP2K and GROMACS, we are not the main developers of CP2K. I just wanted to highlight a recently developed tool within BioExcel, which has attracted quite a lot of interest. And this is the BioExcel building blocks, or BioBBs. So BioBBs, they're Python coded building blocks, and they create a layer of compatibility and interoperability over existing biomolecular tools, not just biomolecular tools, also other tools. And all the BioExcel core codes have been wrapped in BioBBs. So BioBBs enable the easy development of workflows, by basically putting these, multiple building blocks together. So for example, you can use a number of BioBB modules or blocks to prepare and run and analyze a molecular dynamics simulation in GROMACS on a particular protein of interest. And in fact, if you go to the website, so BioBBs are hosted by the IRB. So the link is on the next slide. Sorry, it's not on this one. But there you can find tutorials on how you're able to do this, and there are tutorials for other types of simulations and activities. Or you can also use BioBBs to create your own custom workflow, or even your own building block if you are so inclined. Next slide, please. So bioexcel.eu is our website, and there you can find much more about the types of courses and training and events that we run. There's also information on the use cases that we are working on. You can subscribe to our newsletter. You can follow us on Twitter, on LinkedIn, and you can also access our YouTube channel. Ask BioExcel is our forum for all codes, and this is where we provide our official support and you can also get involved in discussions around molecular dynamics. So you could basically ask any question around any of the codes that we support through Ask BioExcel. The BioExcel webinar series covers lots of different topics, from sort of introductory lectures on how to sort of use codes and set up simulations, all the way through to sort of more advanced topics around best practices and sort of how you get the sort of most of performance out of a particular package if you're running, say, on high performance computing infrastructure. So say there's a lot of stuff there. We've run 54 webinars to date, and these are freely available, and you can find them all through the BioExcel website, and they're all on the BioExcel YouTube channel. I'll say a little bit more about training in the next slide, but I just wanted to highlight as well that we are developing best practice guides which cover the main BioExcel codes, and these best practice guides cover or capture the sort of the know-how which experts use or expert users would follow implicitly when they're using the different codes. So these best practice guides, they're going to be live documents, meaning that they will be continually updated as the software and hardware and their usage evolves. Next slide, please. So I mentioned training. So we've got great software, but to facilitate its adoption, we need to provide great training. And I think based on the feedback, I think we can safely say that we are able to do that. We have been doing that, and we'll continue to do that. BioExcel's built a critically acclaimed training program, and this is based on the BioExcel competency profile, which helps to define what knowledge, skills, and attributes a particular user or researcher in the BioExcel community would need to develop to use our tools. Okay, next slide, please. Okay, so we've established strong collaborations with industry, and this includes large pharma companies like Janssen, Boehringer Ingelheim, UCB Pharma, AstraZeneca, and also a number of small and medium enterprises. And what we're seeing here is a real interest in the application of our tools to some of the sort of the hot areas, should we say, that are sort of sort of hot areas at the moment. So, for example, advanced free energy calculations to guide small molecule design, or information driven docking to help guide therapeutic antibody design or discovery. As part of our work and our collaborations, we've carried out a number of benchmarks between some of our open source codes and commercial solutions, which are used by industry, and shown that the results between the two are comparable. And I think Rossen's got a really nice example in one of the later slides that will provide a little bit more detail. So our codes run on HPC high performance computing resources, which are required for some of these types of pre-exascale calculations or pre-exascale work. And this has been facilitated by some of our partners that manage HPC infrastructure. So, for example, the Barcelona Supercomputing Center, which manages the Mare Nostrum supercomputer, or EPCC at the University of Edinburgh that manages the Archer 2 Tier 1 system in the UK, and the Cirrus Tier 2 system. So these systems, these HPC systems, are all available to academia and industry, and we can certainly help with with information on how to access these if this is something of interest to people in the audience. So to further develop our support and collaboration activities, we're setting up a legal entity. It's called BioExcel Enterprises, and BioExcel Enterprises will be incorporated as an economic association, so think cooperative. And the idea there is that it's a not-for-profit members organization. It's based on the mantra of by members for members, where members can be individuals, can be organizations, consortia. It doesn't really matter. But the important thing is that the sort of the skills and the interests align with those of the BioExcel Center of Excellence. So BioExcel Enterprises will build on the success and the reputation of the center, for example, by working with current partners as members where possible, and also using some of the existing BioExcel branding. But more importantly, BioExcel Enterprises will have the potential to expand the Center of Excellence partner network and thus increase collaboration opportunities. We will also offer services to support these collaborations and increase the quality of scientific research. So if you or your organization is interested in this and your interests align with those of BioExcel, you would like to be part of the BioExcel ecosystem, then please get in touch with us. And at that point, I'll hand over to Rossen.
I can't start my video. I think the host needs to enable it. I can't start my video. Can somebody enable it? Thibaud or Hanne?
Just start talking.
I can't start your video.
Without video. Okay, so yes. Okay, it's coming here. Right. Hi everyone. Good morning. It's a pleasure to be here. It's really great to see so many people joining the event, and congratulations to the organizers for initiating this community. I will continue with a bit more in-depth look at some of the core applications, and specifically look at the HPC aspects of their development, because we are researchers, we are way past the times of using just a single desktop to do cutting-edge research. We do need, and we should make use of the large computational resources that are available to us. So the first application in our core portfolio is GROMACS, as Richard mentioned. You probably all of you are familiar with it. I'm not saying what it's doing, but on the HPC aspects, it's one of the most efficient and scalable codes, open source ones available for molecular dynamic simulations, because largely to the highly tuned kernels written in assembly language, that they make full utilization of the hardware that is presented. Not only that, but modern architectures, supercomputers, they are built of CPUs and accelerators such as GPUs or other more exotic processors, and GROMACS is able to make full utilization of such systems in actually an automated way by load balancing the work given to CPUs to accelerators, because the types of calculations that are more suitable for the different architectures are different. And by this advanced software engineering that is implemented in GROMACS, it's able to show sustained petaflop performance. We have already cases of reaching exascale capabilities. A lot of development work is done in very close collaboration with all of the major hardware vendors, such as NVIDIA, Intel, AMD, ARM, and now the coming European processor initiative. So there are people in the companies who work very closely with the GROMACS developers to ensure that these kernels are fully optimized and tuned. That's why GROMACS has constantly been in the top list of the generally available applications in numerous surveys through the years. It's used in benchmarks, etc. And in some of the plots here, you can see examples of how well the application is able to use hundreds of nodes together with multiple GPU cards, for example, attached to each of the nodes, which is challenging for most of the applications. The other application in the portfolio, Haddock, it's used for integrative modeling, which allows you to do docking of molecules and larger complexes by also incorporating data from experiments such as NMR, IOEM, neutron scattering, and also from computational simulations. You can also incorporate data to guide your docking and improve the scores. So Haddock, in a sense, it is a high throughput, has been used as a high throughput application, but we've shown, and we are using it in BioExcel as part of workflows that are able to make full utilization of HPC resources, such as the example here on Mare Nostrum in Personal Supercomputing Center, running on thousands of cores, doing multiple dockings simultaneously. Those of you who are using it or might be interested, there's a new version coming up with much more improvements and redesign. Another application we developed is an interface that couples QM codes such as CP2K with molecular mechanics codes such as GROMACS, which also Michele, his keynote, was also discussing. This has always been a challenge and a big desire for the communities. There are various efforts in the area. We work with an interface that couples these two very popular applications, and some of the features there include a lot of automatization of the topology conversions, and also there is a focus on compatibility that one can couple other applications as well in the future. And again, on the topic of HPC, it's quite difficult to parallelize such codes on HPC resources. QM usually takes a lot more time than MM calculations, but we've shown how you can get great efficiency on thousands of cores. Now the example with PMX, which Richard mentioned, it's published, you can find more details there, but we've been able to run scoring of almost 500 ligands, and to estimate the free binding energy in a matter of days, and the consensus of the estimates using PMX and GROMACS are on par with commercial software such as FF+ from Shorin, for example. There is big development there. PMX is a great tool that automates the usually laborious process of setting up such calculations. And as a last example, I'm showing how we've used the BioExcel building blocks, which Richard introduced earlier, and with them we have GROMACS and PMX.
Sorry, Rossen, your time is up. Thank you, Richard. And
okay, so just you can visit our website, subscribe to the newsletter, and get in touch with us when.
Yes, thanks for the nice introduction of BioExcel. Then now we move to the next section where you have like some example of project, and the first one will be from Nicolina Sekulic, which is an associate professor at the University of Oslo. She's the group leader of the structural biology and chromatin research group at the Norwegian Center for Molecular Medicine. Nicolina, the stage is yours.
Yeah, just let me share my screen. Yep. Okay, so good morning, everybody, and yeah, thanks for inviting me to tell you a little bit of our research, and especially the one that used MD in this audience. So yeah, I work at Center of Molecular Medicine Norway, and I'm going to tell you the story about activation of Aurora B inbox kinase by auto-phosphorylation. So this is an enzyme complex made of Aurora B and part of NCMM. And this enzyme complex is actually part of a bigger complex that's called Chromosome Passanger Complex. So what I'm going to be talking about is the catalytic module of this complex that consists of two proteins, Aurora B and part of the NCMM. By the rest of the complex, includes localization module that also has borealine, NCMM, and survivin. And so this whole complex is called Chromosome Passanger Complex because it changes its place on the chromosomes and in the cell during the process of cell division. So in interphases on chromosome arms and on centromeres, in metaphase is mainly at centromeres, and then in anaphase and telophase, it actually stays here at the place where the cleft between the two cells is to appear. So it's in each of these stages, this kinase actually has a very important role. So overall, this complex, and especially the catalytic module of this complex, is very important for cell division. And that's why understanding and potentially inhibiting or overactivating this enzyme is something that's considered as an avenue for developing a new cancer drug target. And so we wanted to understand, how does this enzyme complex work? How does Aurora B inbox complex works? And interestingly enough, it actually can exist in inactive state, and it auto-phosphorylates itself in predominantly two positions, one on the loop of the enzyme and one on this NCMM or inbox. And only when it's phosphorylated, it's actually active. So the question that we wanted to ask, or answer rather, is what is really happening at the molecular level to this enzyme when it gets phosphorylated? How the conformation of the enzyme changes from inactive to active. And we were not the first one that were trying to understand this, of course. So people have tried to do it with X-ray crystallography, which would be really cool, right? To see the at atomic resolution, the structure of each of these complexes in different phosphorylation states. However, it was possible to crystallize it only in partially phosphorylated state where the loop is phosphorylated. And furthermore, a part of the inbox that is phosphorylated was not even present in a construct. It took them 10 years to make the protein that actually has this part, but still is flexible in the structure, so they don't see. So obviously, X-ray crystallography is not a good approach to try to understand how the enzyme works and how it's activated. So, oops, that's the wrong direction. So we thought of alternative approach to understand how the enzyme works. And for that, we decided to use to combine experimental and theoretical approach. For experimental approach, we use hydrogen deuterium exchange. And I'm not going to go into the details of how this technique works, but I'm just going to tell you that it's with this technique, it's possible to identify flexible and water accessible parts of the complex. But we also wanted to know structurally at the atomic level what is happening. And for that, we use molecular dynamics and we follow trajectories of the complex. And of course, for that, we needed to be able to have protein in all different forms. So we could obtain protein, and we use actually chemical ligation to make these intermediates. And this was a little project within itself. It was not trivial, but we managed to do it. So we had all the proteins, and we could also measure on all of these different forms. We could measure HDX, and we could also follow molecular dynamics on all the forms. And just before I go into that, I would like to tell you that with the kinetic analysis, now when we have these intermediate forms, we wanted to test them for the activity. And first thing we noticed is that partially phosphorylated enzymes are barely active. So somehow there is a synergy between these two phosphorylations. They are not additive to add to the full phosphorylation. And also in terms of auto-activation, we are sure that these intermediates can fully auto-activate because this is the activity of the fully phosphorylated enzyme. And you can see, and this is unphosphorylated auto-activation, and these are the two others. And you can see that actually one of them, when it's phosphorylated in a loop, it reaches the activity much faster than the one that is phosphorylated only in INSEM. So this was the first clue just from the kinetics. But then we use hydrogen deuterium exchange. And as I said, I'm just going to briefly mention that with this technique, what we are measuring the exchange of the hydrogens in the main chain of the enzyme with D2O. And the assumption is that this exchange is going to be faster in the parts of the protein that are flexible and that are exposed to water environment. And actually, we are the only one in Norway that have this fancy instrument that can do the experiment, and we are kind of proud of it and are happy to collaborate with people on that. But to go back on the project, the interesting thing that we have noticed is that in unphosphorylated enzyme, unphosphorylated enzyme is very flexible, and the lots of parts that are actually important for the catalysis are incorporating deuterium faster, meaning they are more accessible and they're more flexible. We could point out exactly those parts on the structure, but we still, from this data, could not imagine what is happening with them at the atomic level. And so for that, we actually started molecular dynamics in collaboration with Michele Cascella. And these are the results that are actually really, really amazing and gave us a lot of insight in what is happening. So we are starting with this enzyme that has the instamp that is as a coil emerging from the structure far from the active site. So nobody understands how the phosphorylation here can affect the activity of the enzyme. And truly, when we have unphosphorylated enzyme and we do simulation, you can see that this part of Finbox is really exploring many different positions, but it never, it temporarily interacts here with the active site, but not for long. And so I'm not going to play the movie to the end, but you got the idea. So this is how it goes for the end of the simulation. When we have phosphorylation only on the INSEM, something similar is happening at the beginning, but soon this part of the protein adopts a stable interaction with the N-terminal part, and it's again far away from the active site. And it stays here for the end throughout the simulation. Now, when we have the loop phosphorylated, and remember this is the one that is a little bit more active and the one that auto-activates faster, what we observe is that very early on in the simulation, this INSEMP actually going from, remember here, falls down to interact with the loop and stays there for the end of the simulation. Again, I'm not going to play it in the interest of time, but it stays there for the end, until the end. And finally, when we have both elements phosphorylated, again, we will see very soon that there is an interaction between this phosphorylated loop and INSEM. And again, this stays present until the end of the simulation, which is something that we could not predict or appreciate from the crystal structures. Interestingly enough, we also noticed that following these trajectories, that ATP, that the active site is also not really formed, and this is in agreement with our structure, with our data from HDX. We also did analysis of the movements of the protein, and this was very insightful. So that there is coordination between opening, closing, twist, and activation movement that is happening only when both sides are phosphorylated. So in summary, I'm going to say that using the experimental and theoretical approaches, we could figure out the structures of all intermediates, and we could not only understand how the parts of the active site of the enzymes are formed, but also how they are coordinated, making this a completely functional enzyme. And this is something that we can use as a basis for future drug design. And at the end, I would like just to thank people. Most of the experimental work is done by Dyrlie in my lab, and Uda, that was a master student, mentored by Michele. She did MD studies. And yeah, this is our example of how it can work.
Thank you, Nicolina, for a nice example on how to use experimental with molecular modeling and what we can get. Now it is the turn of Geir Hoge-Losse, which is the CEO on Nextera, representing how to use molecular modeling to aid tailored combinatorial protein engineering.
So thank you for inviting me to share some of our work here today. I'm going to share what we've done from a much more applied angle, I think, than some of you others. We're a private company that has a focus on making products, and we depend on quite advanced molecular modeling as part of our path here. And I would like to share in particular one project with you, what we did and how that turned out. So Nextera, in a nutshell, is a company that was spun out from a center of excellence environment called the Center for Immune Regulation at the University of Oslo and Oslo University Hospital. This center has now ceased to exist, but it has given us the opportunity to develop our technology, and we have a focus today on disease-specific target and drug discovery in oncology and autoimmunity. And through the years, we have developed this NextCore platform, which is the basis for what we do, which gives us a unique angle to attack T-cell immunity. Very high level, if you look at the disease target universe, we have never been better able to treat disease than what we are today, but still there are limitations, and those are in particular associated with what we can aim at as disease targets. It's still dominated by cell surface receptors, and many of the targets that we use to treat disease is not directly associated with the disease or specific for the disease. This gives us a very crowded landscape and with few opportunities. Whereas we all know from basic biology that there is a number of targets within the cell. And so underneath the surface of this classical metaphor of an iceberg, which can both be disease specific and be very useful drug targets, but they are very hard to access. So in a nutshell, if you look at the cancer cell, we know that there are really good drug targets on the outside of the cells that are used today with very advanced therapies. So for instance, CD19 and CD20 is dominating the leukemia landscape, and CD19 is also the target for the CAR technology, and later on also BMCA have entered the stage. We have checkpoint inhibitors, but it's not perfect, and we see side effects that is non-tolerable, and in particular within autoimmunity, we cannot use these targets. So within the cell, there is a universe of potential protein targets that could be targeted if you had access to them. And nature have, of course, devised a method to do this by use of HLA molecules. So these are scaffolding molecules that all the cells in the body express, which make complexes with defined fragments from these proteins. And This gives an untapped opportunity for novel targeted therapy, we think, and also many others, which so far have only been accessible through T cells, which is how nature have devised this in the first place. So we have two major compartments of T cells. I'm not going into details of this, but they represent, in essence, how we can acquire the ability to tap the inside of a cell. We know that these proteins, they recognize their ligands in a very conserved manner. And this is taken from a review we had some few years ago, where we pulled out all the structures available at the time. And we see that the MHC molecules here, denoted from a top-down view with the alpha helices, and then a peptide binding group, which encompass the fragment from a potential disease target or a homeostatic protein. And then the TCR comes top down and deciphers this composite antigen. So it has loop structures that then can recognize in a combination of the peptide and the HLA molecule. And this goes for both compartments of the T cells. T cells and TCRs represents one of the immune receptor compartments, but we have a different compartment that is also very important, the antibodies. These are not naturally evolved to recognize the same target. There are examples of how you could get them to do this in the test tube. As far as we know, they are not made by natural processes. And strikingly, what we see here is that they do not at all encompass the same docking onto the target, which there are probably good biological reasons for. Even though many have speculated that making antibodies towards peptide HLA could really bridge this untapped opportunity, it has proven very hard to do. We use a technique called phage display. I'm not going into that, but in essence, phage display is an in vitro method that allows you to acquire large repertoires of, for instance, proteins in the test tube. You can retrieve immune repertoires from donors, integrate them into plasmid vectors, and then you can manufacture big combinatorial libraries on phages. And these can then be used to design selection, for instance, to these targets. And that has proven successful in some cases, and this is what we have done, and this is the project I'm going to share with you. So this is actually a crystal structures from our project, so we know how it ended, and this is a late stage review, so we hope it will be public not in too long, and it was done in a collaboration with Johns Hopkins and Monash. So this is really where I would like to acknowledge our collaborators here. So you can engineer both TCRs and antibodies. I'm now going to focus on the antibody engineering. We would like to do this in this project, not in a cancer setting, but actually in an autoimmune setting. This is where the immune response is actually set into action where it should not. So the immune system starts to attack our own normal tissues, leading to disease. So how could you prevent that? And it is, again, the T cells that is at the core of this interaction. We think that it's possible to engineer antibodies in the test tube that could interfere with this interaction and thereby block the T cells from actually engage into the autoimmune attack. And this was the purpose of the exercise. In all combinatorial or protein engineering efforts, we have this challenge that we need to overcome. We have an ID or a starting point of a fitness landscape that we need to accommodate to actually serve the purpose. It could be that we want to improve the binding of a molecule from being too unspecific or too poor to become more specific or too poor. It could be from a thermodynamics point of view, which comes to stability and manufacturability. Doesn't really matter. The whole point is that working with macromolecules, you very rapidly end into big problems because only a protein of 100 amino acids contains so many possibilities of different variants that you would need to sample that you cannot really experimentally attack it. So you in some way need to condense this very huge number into something that is executable. Luckily, we know that evolution is not working by absolute concrete single units, but it works by epistatic interplay. So there is not one solution to get to this fitness landscape. There are many sequences that can confirm the same function, and the only really challenging task is to get to the high fitness maxima, which are fewer. But it also gives you the engineering window, and this is really where combinatorial protein engineering comes into play if we can use molecular modeling to aid the tailoring of these regimes. And this is what we did. It's on to celiac disease. I'm not going into disease details here due to lack of time, but it's a gut enteropathy, which affects quite many people. It's not very serious for most, but for some it is. And it's also, among others, it underlies one of the more serious leukemias. And there is an unmet medical demand, and we have, through the collaboration at the CIR Center, access to patient material that allowed us to attack this situation. So we, in essence, we did a classical phage display. Again, due to lack of time, I will not go into details, but we did a selection on recombinant proteins that gave us initial binders. And then we saw a need for improving the binding strength and specificity of those binders, and we teamed up with Jeffrey Gray and John Hopkins that is using the Rosetta package for doing these purposes. So very briefly, what we did was to build homology models of our primary antibody sequence, and then we docked those using SnugDock onto the crystal structure of the ligand, which was available. And the aim was here to make targeted libraries. This followed a two-step process. So first we have this initial phase where we dock the antibody structure onto the ligand, and then it's a high resolution phase where we actually refine the whole setup. And if this works now, I might be able, okay, it should be a movie, but see if I manage to get it here. So what you should see here now is the molecules moving, and this is not the rigid body docking, which is one of the beauties of these methods in comparison with the majority of other docking methods, I would say. It's very demanding from a computational point of view, but at the same time, it actually do allow us to move all the parts of the molecules, both in a rotational manner and also in an intra and intermolecular manner, which is really what you need if you want to understand how this could go about. So cutting this a bit short now, it allowed us to focus in on where the parts of the antibody molecule were likely to interact with the ligand and where we could improve them. And then we made targeted libraries that focused this into a sequence space that was actually attackable. And very often forgotten when you want to work with these complex molecules is that so many amino acids that you cannot randomize the whole protein structure. Now, phage display can cope with a huge number, so 1 billion variants is well within reach of this technology, and this is what we made. So we actually made many libraries in this size and sampled those. And again, coming to the end, this went really well. So again, we now know the footprint of our improved binders. The mother clone had an affinity of 240 nanomolar. This was improved down to low picomolar. So this is actually the strongest binder of this type of molecules reported to date, which we find really interesting. And again, we see that epistatic interplay is really working here. So there are many sequences that actually gave rise to really high affinity binders. And we can explain in a detailed manner now, since we crystallized it, exactly what happened. And it was actually an extension of a loop, which massively improved the footprint in a very defined region of the molecule. So the lessons learned from this from us, from an applied point of view, is that modeling is really a powerful tool to build structural hypotheses, and combined and complementing with combinatorial engineering, we can actually then focus these hypotheses into an executable engineering design, which both can accelerate the discovery process and it may improve the success rate. We also have made attempts where we did fail, and the limitations, and this is maybe what we can discuss a little bit later on, is that these processes are really vulnerable to the accuracy of the initial steps. Yes. And I would say that the lack of solvent and still the need for wet lab assistance is limitations that we need to overcome in this space. And by that, I thank you.
Yeah, thank you, Geir, for a very interesting example of how to apply molecular modeling. I would like now to call Gunveig Grenland. She's a research scientist at the Department of Immunology at the University of Oslo, and she will present predicting the formation of immune responses following vaccination in a diverse population. Gunveig.
Thank you very much. I'm very happy to be here to talk to you today. I would first like to begin by saying that, my field is really immunology and vaccine development. And while I have a couple of projects in this area, they are very new. So my starting point today is actually to tell you why am I now progressing into this area. I think we should start with vaccine efficacy as the starting point, because this was indeed my starting point. So what I'm now showing you on the screen is a figure from the phase 3 clinical trial of the mRNA vaccine from Pfizer. More specifically, it's the efficacy. What you can see here is that during the first day after the first vaccination, there actually is very little difference in terms of cases of COVID-19 between those that were vaccinated and not. But then around day 9-10-ish, there is a break, and the vaccine actually kicks into function. And from there on, the gap just increases. And that timeline is very well aligned with the known development of antibodies. So this correlates very well with the timeline that is expected for formation of efficient and protective antibodies. So as you can see from the figure, the vaccine is great, has a high efficacy of 95%. And when I'm showing you this, it's not really because of this 95% efficacy, it's because what happens in those remaining 5%. Because that's actually quite interesting, and it's an area that we don't really know a lot of as of today. Moving back towards influenza, which kind of is more of my original field, then we know that age is one of the factors that would determine vaccine efficacy. What you are now seeing on your screen is that there is a gap in efficacy between people between the age of 15 and 64 and people above the age of 65. So this and the gap is quite significant. And of course, if you went to 80s and with confounding morbidities, the gap would be even larger. And this is known and general for a lot of different vaccines. So clearly, vaccine efficacy is not vaccine efficacy for the entire population. We are all different. Besides age, we know that, of course, the health status will affect how you respond to a vaccine. If you have severe obesity, for example, then, of course, your responses will be different as compared if you are lean. You may also have chronic diseases or other health statuses that will shape the way your immune responses are formed. Sex differences are certainly also a factor for the vaccine efficacy. It's known in general that females, we are much better than males with respect to forming efficient and high titers of antibodies and T cells. But then, of course, the flip side is that that also means we are maybe more prone to getting adverse events after vaccination. There is also a benefit and, well, not a benefit for every situation. Besides these fairly obvious factors that will decide vaccine efficacy, you also have a pathogenic history. That means what kind of antigens have you previously been exposed to, and also your genes will determine how your immune responses are formed, which we expect should have an impact on efficacy. So I'm going to show you, I think this is a pretty cool paper. It's in preprint, or well, BioExcel archives, so it's not been validated and published finally, but it's quite cool. This essentially is a paper that describes how genetic differences can shape CD8 positive T cell responses against SARS-CoV-2. So what they have done here is to make a library of SARS-CoV-2 epitopes that are presented on MHC class 1 on tetramers. And they've used these for selecting out T cells that are specific for the different peptides, and also linked this to genetic background, and then looked at the TCR pairing, as well as the specificity of these T cells, and also what characterizes the effector functionality of the different T cells. And if you see here to your left, it's quite interesting that the gray dot at 180% respectively, way up the large gray dot, this is responses against a particular peptide if you have a B haplotype as your HLA molecule. And interestingly, this peptide is found both in the coronaviruses circulating previously, before SARS-CoV-2, as well as in SARS-CoV-2. So essentially, this genotype has an enhanced benefit with respect to forming broadly reactive T cell responses, CD8 positive T cell responses that could very well have a real impact on how well you are protected against SARS-CoV-2. We don't know this, but it certainly is indicative that there is a difference. If you look at the A haplotypes that are included, these are shown more here in pink. You can see that there is a large difference between those that are formed against people not exposed to SARS-CoV-2 as compared to those that were exposed to SARS-CoV-2, which means that you also get efficient T cell responses with this haplotype, but they are more formed towards new epitopes in SARS-CoV-2 as opposed to boosting things that were already existing. So clearly there is an influence on our ability to form immune responses that are shaped by our genetic difference. So moving into more what we and my group are presently doing. So we are starting with antibodies as our starting point. And what we're now doing is to try to examine epitope binding in polyclonal sera based on a genetic background. And we're using mice as a starting point because it's a simple and clear genetic background. Right now, we're trying to also look at how antigenic previous exposures will influence formation of the antibody repertoire. Right now, we're actually trying to find out how large differences do you have between different viruses in order to actually be able to see the differences at the repertoire level. So here to your left are some of our very preliminary and initial experiments performed by Elias Kjernhage, who is my PhD candidate. He is presently at the Scripps Research Institute in San Diego and performing these experiments there. So we are really trying to determine the antigenic variation needed to look more into the polyclonal response in response to sequential exposure to similar viruses. So this is the situation that you have already for influenza. You're meeting influenza almost every year, well, not the past two, but how does previous exposures influence your antibody responses against the new influenza strain? Or in the case of SARS-CoV-2, how will previous exposures to different variants of SARS-CoV-2 influence the variants that will come in the future? And this is important to know, of course, because this will determine how to handle the pandemic for the future. So what we are now kind of preparing to do is that we will also look to single cell sorting, of course, for these T cells that are specific, B cells that are specific for antigen. And we will link the findings identified in mice to big data for humans to try to validate what we see also in a more relevant context. So I will actually end there. These are some of my collaborators, and going into this project that we are now particularly starting to, I would like to highlight Victor Greif as one of the key partners in that respect. And with that, I would like to thank you for your attention. And next year, I hope to be able to present some really cool data coming out of this project.
Thank you, Gunveig, for the perfect transition, because now for our last speaker, we have actually Victor Greif, which is an associate professor at the Department of Immunology, and he has his own research group that is called the Greif Lab. So Victor, he will present the AI for deciphering the immune repertoire language. Victor, please.
Yes, Thibaud, thanks so much for inviting me to this very exciting initiative, which I agree is really much needed too. I think from the past two presentations from Gerge and Gunveig, we have seen that immunology, and I will also show immunology is in much need for these kind of applications. And I'm also new to this molecular modeling field, so I will just show you some preliminary data that we have and some, and maybe that will give some ideas to participants here for collaboration, which we are always very interested in. So we are very interested in understanding antibody-antigen binding. And so the variable region of the antibody is about 110 amino acids long, and it's the region that is most important for antigen binding. And an antigen can mean a virus, a vaccine compound, etc., etc. But actually, if one zooms into the antibody, it has been shown that an even smaller region of the antibody is actually mostly informative for or important for antigen binding. And this region is on average 15 amino acids long. And so this region of 15 amino acids can basically recognize almost any, so you can find almost, so for any antigen, it is possible to almost find always an antibody that can find this antigen, showing you that there must be, as Gerge also mentioned, high dimensional non-linear space that governs antibody-antigen binding. And I think this is one of the main challenges in immunology, to decipher the rules, the sequence or structural rules that govern antibody-antigen binding. And we don't only want to predict antibody binding to an antigen, so yes or no answer. We would also like to predict where on the antigen the antibody would bind, and this is called the epitope, which was also mentioned before in the previous talks. Yes, so antibody research, as some of you might know, is really key to public health, and it's increasing. So the market size is soon to be projected to reach $300 billion per year. And yes, and it's important for drug design, especially monoclonal antibodies are very popular for cancer therapy and autoimmune therapy, and lately have also become more and more important for treatment of infectious diseases. And yeah, there are a lot of startups for this coming out all over the world, but also in Norway. One problem that we have with antibody discovery is that it usually takes years, up to a decade, to really bring an antibody from preclinical development to the patient. So it takes a long time, it's very expensive, and many of the candidates that go through this pipeline fail. Whereas nature has basically figured out over evolution how to perform this antibody development within a matter of days, as we know, right? So once we have a vaccine or we had an infection, within days or at most weeks, we have a good antibody repertoire against the antigen. And what I think we would like to understand from nature is how is this done, and we think in order to really develop novel machine learning approaches, also to understand these underlying sequence and structural dependencies in the antibody sequence and structure, we will need simulations in order to improve machine learning approaches that help us predict antibody-antigen binding. So what we actually need in the future is some kind of Rosetta Stone that links antibodies to their target antigen. And we have been starting to look at this question 2 years ago, where we looked at then one of the largest antibody antigen structural data sets, which is only 800 structures, so 800 antibody antigen structures, which is very little compared to the potential space of billions and billions of antibody antigen structures that occur in nature at any one time in our body, and so on and so forth. And what has been known up till then most of the time is that epitopes were mostly indistinguishable of other parts of a protein, of a potential protein antigen, if we didn't look at the antibody. And on the antibody side, here in blue, it was known that maybe the binding site is enriched in some sort of, in certain amino acids. But what people have not, or very few people up till then, had looked at is motifs on both sides that maybe could govern antibody-antigen binding. And our hypothesis was, well, if we find a set of those motifs shared across different antibody-antigen structures, then maybe we can find general underlying rules for antibody-antigen binding prediction. And long story short, this was recently published in the Cell Reports, where we actually find a set, or here we call it vocabulary, of interaction motifs that are immune specific. So we don't find them in non-immune protein-protein interaction. This is a quite small set, and it's shared across antibody-antigen structures that are very different, so different antigens and different antibody sequences. And we could also show that this motif set then enables predictability of antibody-antigen binding. The problem, as I said, that this data set is very small, so we cannot really use it to develop novel machine learning approaches. Therefore, we are now currently developing, hopefully soon putting out as a preprint, a simulation framework for in silico 3D antibody-antigen binding complex simulation. And using the simulation framework, and we'll go very fast here, we can now, instead of having experimentally 1,000 crystal structures, we have now 1 billion crystal structures. These recapitulate biological complexity to a very large degree. And with this data set, where we have now a structural paratope, epitope, and affinity information, we can then start to really benchmark machine learning approaches with respect to prediction accuracy, but also interpretability. Yes, and this is just for the experts that might be listening in. We employ here an exhaustive docking technique to really find the optimal paratope epitope structure for each antibody antigen that we are looking into. Yes. And what I think is really important, that the simulation framework preserves major part of the complexity of antibody-antigen binding, which is very different antibodies can bind the same antigen, and very different antigens are bound by very similar antibodies, which was also mentioned in the Argus talk. Yes, so now we can really, in an unconstrained fashion, in silico explore antibody specificity prediction methods. And we have further applied this framework also to look at, okay, can we use machine learning in a generative fashion for finding new binders? And I will not go into detail, but a long story short is that we have shown here that, or we have provided a proof of principle that deep learning models can actually learn 3D binding rules from 1D sequence data. And the framework that we have developed here basically, we think, paves the way for fit-for-purpose design of antibodies with epitope level precision, and importantly also takes into account developability parameters, which are really important, and antibody designs such as, so these are physical chemical properties that are important in the later stages of the clinical testing. So we are providing here a proof of principle that antibody-antigen binding is predictable. And we are now looking into in vitro validating our approach. And we are also looking for collaborations and help with actually further refining our simulation framework with docking approaches and molecular dynamics approaches. Yes. And with that, I would like to thank my team, our collaborators, our funders, and thank you for your attention.
Thank you, Victor, for the nice presentation. Very interesting. So this closes our last the session for the small project, and we move now to the fishbowl discussion. And I will call Naji. Here you are, and I will leave the floor to you and the moderation. Thanks everybody for your attention.
Okay, thank you everybody, and I'd like to first say thank you to everybody who's joined and presented. For this first, I call it NORCAM. It's not, as Thibaud said, the name isn't decided yet. This is what we want to do in these last 30 minutes. We've seen what molecular dynamics is, and molecular modeling is, what it can help. We've also seen indications of what the limitations are. In a sense, pointing towards the future and how it can be improved, basically. We've seen example tools and example projects. So that's what we saw up to this point. I'm not going to, I have a full presentation for the summary, but we won't have time for that. Let's see how much we can do in the limited time. So what we want to do now is really ask you a few questions related to the future of this community or this society, if you want, if you see any difference between a society and a community, what the name should be, what the organization should be, what kind of services this community should offer, and all that stuff. And I'm going to show you the questions, and I'm going to invite some of our panelists to say a few words, their mind related to the questions, and try to answer the questions. But we have also a post-seminar form. You will be able to get it probably during the day today, right after this presentation, this seminar, and latest tomorrow or in a few days. And please fill that in so that we know what we're going to be doing, what we're going to be focusing upon. These are the questions, and with a little suggestion. It's as Thibaud said, our aim is to promote molecular modeling and simulation, help motivate scientists, especially the young ones, PhD candidates, postdocs, and all the others, as well as the industry, trying to put together tools in the biomedical space. And we want to be able to create an open and benevolent network of not only professionals. The use of that term is very conscious. We want to set up a network of friends that can cooperate across. There is no such society. Many countries have these this kind of a society, either related to molecular dynamics or molecular modeling, simulation, computer-aided molecular design, whatever you want to call it. We don't have one in Norway, and it is becoming more and more important because it's a tool that helps. So that as a suggestion and background. Our questions are: was the event useful? In what way was it useful? Do we want a Norwegian computer-aided modeling design society or community? And society is more scientific in a sense. It doesn't really matter. Community is maybe more a community for all professionals, not necessarily only the academical ones. What should the purpose of this society be? What should the organizational form be? Very important questions. Would that be only professional, only scientific, or a mix? And should it be with paying and non-paying members? Should it be with an elected or an appointed board? Maybe rotating every year or second year or third year, whatever. And what kind of services would you want from such a society or community? Examples: high-performance computing was mentioned, computing resources, easy access to them, maybe advisory services support like what BioExcel went through, maybe a regular annual or biannual congress where people could go and meet and be friends, establish a network, maybe with PhDs and the scientific people presenting their work and meeting the industry, or maybe workshops or training sessions, or a summer school was mentioned. We had a meeting with the French version of the society yesterday, etc. So there are many, many alternatives. So what I am going to do is now call people to, I'm just leaving this so that you see the questions and the goal. I'd like to invite Michele first, a couple of minutes to say what he wants to say. Michele?
Yes, thank you. Well, I mean, I think it was very useful. Actually, thank you for organizing that again. Okay, I can share my video now, I think.
I have to stop sharing. Okay, go ahead, share. Go ahead.
I don't want to share the camera. No, please keep the question so I keep my...
Okay.
So that I don't lose the focus on the points.
Okay, I'm sharing again. Yes.
Okay. Yes, so I think it was useful, and it was particularly useful to recognize and identify that actually in our country and in our area, there are many players in the actually active in modeling and in simulation. And actually, there is a community that does both development and applications. And of course, I think networking in this community is very important, because the feeling is that we are largely invisible and covered, and by more organized maybe groups, more visible groups. I thought from a chemical perspective, clearly the quantum chemical community is very strong, very active, and dominating, for example, the regional chemical society meetings, where I am practically almost always the only one presenting molecular modeling, while 90% of the speakers or the other people that attend typically presents just quantum chemical models. And this reflects also on lobbying and networking. I think is useful because this is also reflected often in the panels that compose the NFR, where often you have people that are actually not expert in molecular modeling that review molecular modeling applications, and then of course bias in the. If we are visible and present in numbers as a community, then we can also have a sort of put pressure so that maybe also NFR panels where experienced evaluators are called in there are more on our community and they can review better our projects.
Mm-hmm. Yeah.
So I think, I mean, this is very useful. So networking, I think, is very important. In terms of the form, I don't know if we are at the level that we should jump into a formal society. Maybe we could start with a more informal network, and then one can, in a sense, it's a trial and error. I mean, one can start building something, then see how it goes, and then expand. I don't have strong opinions on that, actually.
Okay.
And of course, I mean, in terms of servicing, well, this depends, of course, a lot on the people that enter in the community and then wants to be active in the community. Clearly, in the moment, it would be very nice if we could actually bring in both, for example, I see several experimentalists and then people will work with data that now feel the need of integrating modeling in their research. And then, of course, there are more theoretical groups that instead will be more into developing also an application that can actually, so creating anything that can favor the information flow and the technical flow and help each other. So I think this is the kind of things we know of. I understand, for example, there is a lot of many things already happening in BioExcel, that this could be probably immediate interest for several people who just want to learn how to run, for example, use GROMACS and have a good training and other things, but there are also more advanced methodologies where there is more, say, on a research or a development level that would be nice maybe to, if one is in direct contact, to also transfer to people that instead are more interested in applications. Yeah.
Thank you. Thank you. It was concise and it's under 5 minutes. That's very, very good. So we can have time for other people as well. I'd like to ask Geir Åge if he's still around. He is one of the few in the panel from the industry. Geir Åge?
Yes, thank you. First of all, I think that this initiative is great. Stating the obvious, I think we often forget as focused scientists with too limited time that you don't know what you don't know. And that is maybe in a nutshell what this is about. And a good way to approach that is to talk to people. But you don't know who to talk to and when to talk to and what about necessarily. So to arrange a meeting place like this forum could be, I think is a good way to start. I don't know the answers to exactly how this should be organized, but it needs, of course, to be focused into something that you find interesting, again, due to the competition of time. Already now, I've been learning a lot of things that I was unaware of. For instance, the BioExcel BB package and these type of things seems like great initiatives, and which again just spurs that you need information. I think that one needs to have a focus on how this should be arranged if you would actually like it to work between academia and industry. As we as a commercial unit, we cannot operate by the same transparency, for instance, like all of you academia, for obvious reasons. This is not a problem, it just needs to be tackled. And there are several Norwegian initiatives that has failed to embrace this notion. I think this is common knowledge in US and in Central Europe. So I would really also urge those that are going to organize this to take this into account, because we need to have clarifications regarding simple things like how to share information and data, and also how to tackle IP if that is an issue. But not making problems. I think that to generate a meeting place for this, I'd really like to. We needed to go to the US and to Australia to find collaborators to help us solve our problems.
Exactly.
I don't think that necessarily was necessary, because I think that that there are people here in homelands that actually could help us out, but we don't know them. And what I do know is, for instance, when it comes to very precise hurdles, access to infrastructure that could help us do this notion here in Norway is prohibited due to legislation in Norway. So for instance, we as a company are located in Forskningsparken, but we cannot access the high performance computing facilities, for instance, of the university. Which of course effectively prohibits us from actually do the work. So we need to go abroad. So this is also something that this community could actually facilitate, how to bring down barriers to facilitate actually the interplay. This could be an endless discussion, so I think I'll leave it with that, and also it would be interesting to hear other thoughts on this.
Thank you very much. It's I believe this high performance computing stuff, I have been exposed to the challenge that you just mentioned, so that's one of the reasons why it says computing research as a suggestion. Basically, we should talk about that, and we need to remove the barriers both between industry and academia and in terms of access to certain resources like this. All right, so I have two opinions. I need more, and I would like to ask Gunnveig if she is around.
I would like very much to thank you for organizing this. From my perspective, it is really important to know what's around in terms of abilities in Norway, because I have a lot of research questions to which the toolkit I presently have available is limited. And to identify which technologies exist that could help me solve the questions that we are presently posing, that would be very helpful for me, because as of today, the strategy is more to, Google, use PubMed to try to find people internationally who can do this, and then potentially bypassing people in Norway who could be even easier and very relevant collaborators. So for me, that's an important perspective.
Mm-hmm.
As to the form of organization, I'm not really concerned with payment or not, as long as the threshold is set so low that this is not prohibitive with respect to having a participation, both from people who know they are interested, but also people who think they might be interested to then get knowledge on what actually is going on. So in terms of services, I think, as you have suggested, workshops, this event, summer schools for PhD students or master students, training sessions, would be very relevant, but would also maybe require that you have at least some funds available. So therefore, maybe arguing why you should have a small symbolic fee, but it has to be low, I would say, if going to be there. Yeah.
Okay.
Thank you from me.
Thank you for you, and thank you for everybody. I think I have about a couple of more minutes. If I can just get Victor to just follow up. The work that you're doing is
Victor had
close.
to run.
Okay, Victor had to run.
But Rossen raised his hand.
Yes, Rossen, please.
I can add to that. So these questions, they're very important. From my experience with the working dogs over the years, we've kind of worked on similar ones. This is a lot of effort. It will take months to answer them. What was just mentioned, I think it's important to know, do you have in mind some funding schemes that can support the development of this community? Because, for example, running irregular get-togethers is very low effort. It has a lot of benefits, but doing something more will require time and possibly money as well. So if there are funding schemes which you might be able to tap into, then the proposals need to be aligned with this. But this will take a lot of work. It can't be answered. I think it will take months until this is fully clarified.
Nicolina would like to add.
Yeah, go ahead.
Basically reiterate the same thing. But I think that the first step would probably be to have some kind of a website, and maybe, you know, this meeting and like at least once a year or once in 6 months is something like a small meeting like this, and with a list of participants and you know, programs, so that people know that those are the people in the community that are interested and are doing something. And I think that would be the first very low barrier step to make it visible, at least somehow. Yeah, that's just my 2 cents.
Okay, thank you very much, Nicolina. Thibaud, who was the person who was going to?
Yash Pardasani, but I don't have the right to unmute that person. I'm not sure if he's
And he has the microphone now.
Yes.
Yash, go ahead.
Hi. First of all, thank you so much for organizing this meet. And it was really wonderful. I'm currently working at Teesside University on metabolic data modeling and visualizations. And attending this, it was a great pleasure knowing about all these things, working on the industry. And I'm currently new to the industry about modeling and these all things. I've done my bachelor's in life sciences and my master's in bioinformatics. And I'm currently working as a visiting researcher at Teesside University. So we are handling with all these data sets, and I was more interested towards this kind of work, like modeling and atomic simulations and all these things about antibodies and antigen reactions, and particularly with the pigment pathways as well that are present in different organisms, especially the species which are there, which we haven't explored much. Recently, I saw one more expedition like this, one more seminar like this on oceans expeditions. And they were talking about the same things, that there were a lot of things that are unexplored right now. And attending the seminar, I mean, it has opened a lot of ideas towards me. Thank you so much for organizing this. And I was wondering, like, if it is possible in future, by any chance, is it possible to be a part of a mailing list or something about this committee or webinar to know more about it, or if there are any opportunities available for volunteering or participation?
Well, Thibaud, would you like to answer that?
Actually, so regarding the mailing list, yeah, we can set up something. I mean, this is possible. So we're gonna see what is the best way to actually create a sense of group on maybe website. I mean, we have to decide. Like, we have to see the effort on the number of people interesting to join. So we will see that.
Okay, well, I think I'll just say a couple of words, and then we're past 11, so I'm going to wrap it up.
Not I think. The 20 seconds comment from my side.
Yes, of course.
Yeah, thank you. To see that the initiative made by Thibaud and how he has now joined the first working group, and also this first meeting, how well it is received in the community. And this is really a proof, Thibaud, on what you saw, that there's a lot of people both with competencies and with needs in the ecosystem, but they do not have any platform to find each other. So we are really excited to see how this will move forward. And from the cluster side, we will also continue supporting you in the early stage, which is, as we all know, having tried to start communities of different kinds, always the most difficult time. So support on any kind, as we have done now also, we will continue to discuss with you.
Yeah, thank you, Mette, for the words. Yeah, thank for everybody to be there. It's true that, I mean, I'm very happy about the response of everybody I've been talking to. Everybody said that it's needed. Israel, I've seen other countries that is well developed, is developed. I mean, Germany, France, Canada, USA, UK, they haven't started doing that. There's nothing in Norway, so there's definitely some potential there. And I'm very happy about that we are supported by the Life Science Cluster that give us visibility and as well connection. This is how I met Naji. So without that, I mean, we wouldn't be there. It would just be, I don't know, a small seminar with like 5 person. And it's true that we need to connect with everybody on maybe as Björte work in the chat, that maybe we need to find like some fun, maybe to organize some courses, and then slowly we just get the word out and people will see the interest in it, because we need to raise the awareness and to bring back together theoreticians and experimentalists together, because this is the only way we can create a synergy to make good science and good innovation.
Yes, we're about to be finishing. It seems we have received a good deal of answers, but for those who have not been talking, please, the same questions basically are being sent in a link in a survey. Please answer as much as you want. You can put your name, or you can answer anonymously, or you can ask questions basically. So it's perfectly okay to be anonymous as well. So far, what I am hearing is that this event was useful, and it seems we need something like a community, or at least a hub that gets the information, gets people to talk to each other and sort of enables information. We are for. Rossen warned us that it's going to be a lot of work and we will need some funding. We will have to sit down and talk about that. But Bjarte Aarvold, he wrote that there is support from Norwegian Biochemical Society for arranging courses and such events, I suppose. We probably will be able to find some means. One of the things that we know from other such societies is that they have a very, very symbolic or no fee membership, but they take some money for the conferences and courses they arrange, and that could be one of the things. But please answer. We are still discussing. We hope to continue these, and we'll come back with some sort of a plan, I hope. So, yes, any last words, Thibaud or anybody else? Actually, I am going to round it up.
I'm done. No, I'm happy. No, I'm very happy. Like the last month of networking and discussion, I mean, I've been fruitful and looking forward to the development of this community. It's really needed. I hope to achieve like something like France. I mean, we were discussing with them yesterday. It was like very friendly atmosphere. This is what I want to recreate here. We need each other. We have a small country, and I know there is a lot of capabilities. So we should definitely stick together, have a drink, have a seminar, whatever, and then move on.
I hope we'll get to the point where we can
actually
have the drink together. That would be very nice.
Yeah.
All right. Iselin, take over. I'm done. And there is nothing more, so you can actually finish.
All right. Yeah. Thank you all.
And thanks again for the Life Science Cluster. Yeah.